everyone, I'm Sandhya. I'm your botany lecturer. In the last video we have started the chapter sexual reproduction of flowering plants. Then we have discussed the male reproductive structure, the stamen, anther, microsporangium, pollen grain, everything about the male reproductive structure we have studied. In this lecture we are going to deal with the female reproductive structure. The female reproductive structure. The female reproductive structure is the female reproductive structure of the flower of a flower is the gynoecium. Gynoecium part. Okay, the gynoecium part, or it is also called as pistil, or it is also called as carpel. Carpel. It is the innermost wall of the flower. Then based on the presence of number of pistils and carpels, if one carpel is present, it is called as monocarpellary. It is called as monocarpellary if one carpel is present. If many are present, it is called as multicarpellary. Many are present, many carpels. So, if many carpels are present, we might be getting that if they are free or fused. So, if they are free, if these many carpels, many pistils are free, such type of conditions is called as apocarpus. Apocarpus condition, apocarpus ovary, which is present in, apocarpus is present in Annona and Michaelia. Annona and Michaelia. If the carpels are free, then if the many carpels are present in the gynoecium part, then if many carpels are present, if they are fused, if they represent as one, such type of condition is called as syn. Syn means fused. Syncarpus ovary. So if the carpels are fused, it is present in hibiscus and papaver. Hibiscus and papaver. Okay. So if we study about the parts of gynoecium, pistil or carpel, it has three parts. It has three parts. Okay. Like this. So the basal lower portion is called, the basal bulged lower portion which is attached to the thalamus is called as ovary. Then the portion which is above the ovary connecting the stigma, or the part which is present in between stigma and ovary, or the elongated portion below the stigma is called as style. This is called as stigma. The tip of the gynoecium is called as stigma, or it is the landing platform of the pollen grain. So the landing part of the pollen grain of the landing platform of the gynoecium where the pollen grain is going to land is called as stigma, and the part which is present below the stigma is the style. It is an elongated part. It is like long tube like. Then below the style is the bulged portion which is somewhat attached to the thalamus. This bulged portion is called as ovary. So these are the three parts of the carpel or pistil, or you can say gynoecium, Then so this particular ovary is going to have a ovarian cavity. That ovarian cavity is called as locule. Okay. Then in this ovarian cavity, many placenta will be there. To the placenta, the ovules are going to be attached. The ovules are nothing but the megasporangium. Which will be having the megaspore mother cells. These megaspore mother cells will be producing the megaspores. That megaspore will be developing into the embryo sac, which is the female gametophyte. In that one, the female gamete is going to be present. In detail we will study what it is actually. So the ovarian cavity is called as locule, and in this locule the placenta is present, and to this placenta the megasporangium or ovule is attached. And the placenta is present, to which the megasporangium or the ovule sporangium means singular, sporangia means plural. So ovule is going to be attached. Sometimes in this ovary, maybe one ovule may be present, or many ovules may be present. So one ovule is present in wheat, paddy and sunflower, where inside the locule, inside the ovary, only one ovule is present. So one ovule is present in wheat, paddy and sunflower. Many ovules are present in many of the angiospermic plants. Like you can say watermelon, orchids, orchids and papaya. Papaya are going to have many ovules. So this is about the just introduction of the megasporangium. In detail we will study about this megasporangium ovule. Okay, so the megasporangium is the ovule. Megasporangium is the ovule. So this ovule is a small structure which is attached to the placenta by means of a stalk called as it is attached to the placenta by means of a stalk called as funicle. Let us see the diagram for this. Okay, so this is the diagram of the ovule. Okay, like this. Okay, so the part which is in between the funicle and placenta is called as hilum. So this part is hilum. The stalk is called as funicle. Okay, the stalk is called as funicle. The body of the ovule fuses with the funicle. At the region called as hilum. And the hilum represents the junction between the ovule and the funicle. Then each ovule will be having one or two integuments. So these are the integuments, one or two. These are integuments. Sometimes these integuments may be one or maybe two or more. And these integuments, they are not fused at the tip. They form an opening which is called as micropyle. This is called as micropyle. These integuments are not fused at the opening of the ovule. That opening, the pore of the ovule, is called as micropyle. Then opposite to the micropyle, this basal portion is called as chalaza. This portion is called as chalaza. Then these two integuments are enclosing a tissue. This tissue is called as nucellus. It is a nutritive tissue. Okay, this portion is called as nucellus, which will be enclosed in the embryo sac. Okay, so this is nucellus. Then the nucellus is encircled by the one or two integuments. It is not at all covered by any of, it is not at all covered, no integuments are present in Loranthus. Example, in Loranthus, there are no integuments at all, just the nucellus is present, no integuments. These are all neat questions, you have to remember. Okay, in Loranthus there are no integuments at all. Then the ovule is not covered by integuments. Then in Helianthus, that is sunflower species, and Datura, in Helianthus and Datura, the ovules are covered by single integument. Only one integument is present. Okay. Then in monocots and polypetalous, petale members will be having two integuments. Majority of the angiosperms will be having two integuments. Later, after the fertilization, these integuments are going to form into seed coat anamata. The seed coat is the covering of the seed. That outer covering is called as testa, the inner covering is called as the tegmen, like that. So these integuments are going to form into the seed coat. Okay, so if you see this outer covering, the outer covering is called as outer integument. Outer integument. The inner one is called as the inner integument. Later, after fertilization, these integuments are going to form into testa and tegmina, which is the seed coat of the seed. Okay. Then coming on to the nucellus, it is a reserved food materials. The cells of the nucellus is having the reserved food materials. It's a nutritive tissue. It is a nutritive tissue. Now coming on to the types of ovules. What are the different types of ovules? And this nucellus tissue, after few divisions, it is going to produce the embryo sac inside. We'll discuss. Okay, we'll discuss in the next one. Okay, we'll discuss in detail afterwards. But before that, we'll discuss about the types of ovules. First, types of ovules. The Megasporangium. Types of ovules. The first one is orthotropous ovule, which means like this. The ovule is a straight ovule. The ovule is a straight ovule. The ovule is a straight ovule. Okay. Okay, where the micropyle, chalaza and the funicle are in straight line. This is the straight ovule, which is called as orthotropous ovule. This type of ovules are present in mainly in polygonium. Okay, orthotropous ovule. So on the same vertical line, the micropyle, chalaza and funiculars are present. Then so coming on to the next type of ovule, it is called as anatropous ovule. The second one is anatropous ovule, which can be seen in sunflower and datura. So how it is present? Simple. This is straight ovule, no? It takes a curve of 180 degrees such that the micropyle, I'll draw. Like this it is there, no? Like this, such that such that the micropyle and the funicle are side to side. Means such the straight ovule has taken a turn of has taken a curvature of 180 degrees anamata. It has taken a curvature of 180 degrees. Okay, so the ovules of sunflower family are inverted. Okay, this is a straight ovule. Orthotropous is a straight ovule. This is an inverted ovule. This is an inverted ovule. Okay, this is an inverted ovule. Then, and the micropyle lies close to the funiculus. Funiculus as a result of 180 degrees curvature. Such type of ovule is called as anatropous ovule. Coming on to the third type. Campylotropous ovule. Okay, this is a characteristic feature of bean family. That is in Pisum, Dolichus lablab. In all leguminosa members, members of bean family, the campylotropous ovule is present. What it is? It is nothing but the body of the ovule takes an angle of 90 degrees. Here it takes an angle of 90 degrees. Okay, it is such that the body of the ovule is placed at 90 degrees to the funiculus. Okay, so here like this. And here the embryo sac takes a curvature. It is slightly bended anamata. It is slightly bended. Okay, this is the funiculus. Okay, funicle. The body of the ovule is at right angle, isn't it? It is at right angle. And here the embryo sac is slightly curved anamata. That is a characteristic feature of the bean family. Such type of an ovule is called as campylotropous ovule. So in campylotropous ovule, the body of the ovule bends in such a way that the micropyle comes towards the funicle and the embryo sac is slightly curved. And these ovules are called as campylotropous ovules. So this is about the types of ovules. Coming on to the megasporogenesis. How a megaspore, how a female gametophyte, how the female gamete is formed, we are going to see. Formation of female gamete. The female gamete is formed from female gametophyte. The female gametophyte is formed from the Megasporangium. Okay. So now we are going to deal with Megasporogenesis. That means formation of megaspores. formation of megaspores. From where? From megaspore mother cells. Megaspore mother cells. That is MMC, which are present where? In Megasporangium, which is ovule. That is in the nucellus. Okay. The formation of the megaspores from the megaspore mother cell is called as the megasporogenesis. So one of the megaspore mother cell will be forming into one MMC is going to undergo the meiosis or the reduction division, which will be resulting into 4 megaspores. Okay, 4 megaspores. So this megaspore mother cells are present inside the nucellus. Okay, so let us draw a diagram now. Take for example, this is the nucellus, isn't it? Like this, many one megaspore mother cell is there. Okay, this is the one megaspore mother cell. Like this, many cells are present. This is one of the one megaspore mother cell. One MMC in nucellus. It is going to undergo meiosis, so hence it can be shown as like this. Okay, so here it has become megaspore tetrad. 4 are there, no? So megaspore tetrad. Okay, so in this megaspore tetrad, this particular nucellus is at the micropylar end. At the micropylar end, it undergoes the meiosis. Meiosis, 4 megaspore tetrad. From this, we will take one of them. Okay, we will take one of them. Okay, we will take one of them. See here, one of them. So here, one of this nuclei, the nuclei of one of the megaspore, the functional megaspore, it undergoes mitosis. Then that nucleus will move on to the polar region. So one mitotic division has taken place in the nucleus. The two nuclei has moved to the ends of the megaspore. So here the nucleus has undergone. Now the cell didn't divide. It is a free nuclear division. The nucleus has undergone the mitosis division. Then these nuclei will again undergo. I'll write down here. Okay. From one mitosis. Then it will again undergo mitosis. As a result, 2 will be formed. Then it will again undergo mitosis. As a result, okay, 8 will be formed. Second mitosis, third mitosis. Okay, then one of the nucleus at the polar ends will move towards the center. Okay, so here like this. So these are all free nuclear division. Here the cell wall formation didn't take place. Now after the formation of this particular 3 cells at three cells at the polar ends and 2 cells at the center, the cell wall formation takes place. Okay, so if we draw a mature, so this is an embryo sac, initial one. If you draw a mature embryo sac with the cell wall formation, it will be like this. This is at the chalazal end, okay? Chalazal end. These are the cells now. These are cells. These are the nuclei. Here the two cells will be embedded in the, two nuclei will be embedded in one cell. Okay, this is two nuclei in one cell. This is called a central cell. Then here like this is the egg cell. Okay, so the three cells which are present at the chalazal end are called as antipodals. The central cell is also called as polar nuclei, which are having two nucleus. Then these two cells are called as synergids. Just below these two synergids, a finger-like structures will be present, which are called as filiform apparatus. Filiform apparatus. Above the synergids is the egg cell. This is our female gamete, which is embedded in the embryo sac. This is the embryo sac, the female gametophyte. Embryo sac. What it is? It is the female gametophyte. Okay, female gametophyte, which is having the egg cell. Okay, as this egg cell is formed from one megaspore, such type of embryo sac development is called as monosporic development. It is called as monosporic development. Okay, it is called as monosporic development, as it is developing from one megaspore. And these are having how many cells? 1, 2, 3, 4, 5, 6, 7. So it is 7 cells. The embryo sac is 7 celled. How many nuclei are present? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleate. 8 nucleated embryo sac. So the embryo sac is monosporic development and 7 cell and 8 nucleated one. 8 nucleated one. This is about the female gametophyte. Now the egg cell is ready. Okay, so again a brush up of this embryo sac. This embryo sac is embedded in the nucellus. The nucellus cells are the Megasporangium. So one of the cell of the nucellus at the micropylar end, the megaspore mother cell, will undergo the meiotic division. As a result, 4 megaspores are formed. From those 4, only one is functional. That one megaspore, the nucleus of one megaspore will undergo the mitosis. As a result, 2 will be formed. Then again the second mitosis takes place. 4 nuclei are formed. Then third mitosis, 8 nuclei are present. From those 8 nuclei, 2 of them will move towards the center and form a central nucleus, polar nuclei. Then the cells which are present the cells which are present nearer to the chalazal end, the 3 cells are called as antipodals. And the cells which are present near to the micropylar end, these are called as egg apparatus. The egg cell plus synergids is called as egg apparatus. So the 3 cells which are present at the micropylar end, having the egg cell and the synergids and the filiform apparatus, is called as egg apparatus. So as a result, it is 7 celled and 8 nucleated, as it is forming from one megaspore. It is monosporic development. And what is the function of this filiform apparatus? These finger-like projections are useful to guide the pollen tube carrying the male gametes into the embryo sac. The filiform apparatus will be guiding the pollen tube to enter into the embryo sac. The synergids also through the synergids to enter into the egg cell. There the syngamy takes place. Okay, so after fertilization, what happens? This synergids, this antipodals, they all degenerate. The egg cell will be developing into the zygote to embryo. The polar nuclei will be developing into primary endosperm nucleus or PEN, which again develops again after fertilization to develop into endosperm. Later on we will study. So this is about the formation of the female gametophyte. So now in the last video we have studied the formation of male gamete, and here in this video we have studied about the formation of the female gamete. In the next video we are going to study about the transfer of the pollen grain from one flower to another flower, pollination. Thank you.